Welcome back. So this section has to do about organelles that are not part of the endomembranous system. And so we're going to start with cytoskeleton. In other words, what is giving shape and strength to the cell itself. And we have three different categories of cytoskeleton, known as microfilaments, microtubules, and intermediate filaments. And so the reason why I have this box here is because when you were studying and when you were reading, you need to make it an active process. And so here's what I want you to do. I want you to stop for a second, because I would do this in class if we were meeting face to face, and sit there and try to predict between these three types which is the largest and which is the smallest. Just look at their names and try to figure it out. And then once you think you have it, and commit yourself, take a pen and write it down so that you have committed yourself. And then we'll come back and we'll talk about them. Okay, so most of you at this point figured out that micro is going to be smaller than intermediate. So you thought the two micro words would be the smallest and the intermediate would be the biggest. Okay, well, I'll give you that a microfilament is going to be smaller than an intermediate filament. They make sense. They both have the word filament, and the only difference is between micro and intermediate. So then you have to look at the word tubule. What does tubule imply? What is a tube? It implies that it's hollow, right? Which means it has to be bigger than a filament. A filament implies that something's like pretty small. So even though tubule has the prefix micro in front of it, a microtubule is going to be bigger than an intermediate filament. So all of these exist in our cells. So microtubules are the examples of these blue tubes and things. Think of them like the dividers on the street. You have to go all the way down to the end and then come back in order to get to where you need to go. You can't cut across them. Anyway, I'm going to go through all of these one by one. So let me just go ahead and talk about them. On this slide, I do have written examples in the smaller print underneath each of these, just in case you're looking at this for review later on. But I talk about these as we're going along as well. So if you got it wrong, don't worry about it. Scientists aren't right all the time. In fact, that's what the whole point of experiments are. 90% of experiments give us the answer we were not expecting. And that's what science is, figuring out what the truth is. All right, so let's start with microfilaments. So microfilaments are made up of little proteins, and the proteins that they're made up, an example of a microfilament that we're going to be talking about in the human body is something called actin, which you don't have to know for this first test, but is going to be super important when we're talking about muscles. So we'll learn a lot more about it later. But in addition to the actin for muscles contracting, we have these microfilaments in all of our cells. Lots of it just below the plasma membrane, as well as surrounding the nucleus, and they provide some kind of mechanical strength to the cell. And they're going to be super important when cells are dividing at the end of mitosis, that process known as cytokinesis. If this is a white blood cell, it's not, but if it was and it needed to move to engulf a bacterium, that's what allows it to engulf the bacterium. And as I alluded to, actin is one of the main proteins involved in muscle cells contracting. In contrast to a microfilament, an intermediate filament is thicker. So think of like a braided rope, a twisted rope. So it has more strength to it. And an example of this is keratin. And keratin is a protein most of you have heard about because keratin's in your hair. Okay, we got keratin other places in our body. They're going to be attached to cell junctions known as desmosomes, which we will talk about in the last video about the cell. So we'll get back to that shortly. And the third type of cytoskeleton is a microtubule. So as you can see, yes, it's hollow in there. And as I alluded to before, organelles can attach and move along their pathway to cruise in the cell. In fact, there's a little 8-minute video of this you can watch, and maybe I'll post the link, or if you're interested, ask me and I'll give you the link for this. They're also going to be found in some of our organelles related to the cell, our cilium, our flagellum, and our centrioles, which I'll be talking about right now. So the centrioles, every cell has 2 of these. These are these blue structures, and if you notice, they are perpendicular to each other because they're always perpendicular to each other. And each centriole is made up of 9 groups of triplets of microtubules. So what does that mean? That means there are 3 and that there are 9 groupings of them. You don't need to know that there's 9 groupings of 3 of them. You just need to know that you have a whole bunch of them going around in a circle. And these centrioles are going to be sending out microfilaments, which are going to be attaching to the chromosomes during the processes of mitosis and meiosis when cells are dividing, forming that spindle. So super important for cell division. We'll be talking about them again when we talk about the reproductive system. So until then, if the cell, we're not going to be mentioning them. Another place found on lots of cells, on lots of organ systems, is something called a cilium. So that is the singular form. So one thing you may have noticed by now is we always use singular form, okay? Because when we're pointing to something, we're asking you to name it, we're pointing to one structure, asking you to name one structure. So you need to know singular forms as well as plural. So the singular form is cilium. And a cilium basically is a projection from the surface plasma membrane, and it sticks out, and basically it's plasma membrane surrounding a bunch of microtubules. And so these are the structure. And so a bunch of these together are called cilia. So you won't hear me talking about cilium, you'll hear me talking about cilia because that's the plural form. So for this, I want you to stick your arms up in the air. Everybody stick your arms up in the air. And in class, I wait until everybody sticks your arms up in the air. So just humor me and stick your arms up in the air because it will help you remember this. Your body is the cell. Your arms, each one is a cilium. Now I want you to move your arms back and forth like this. So keep on moving them back and forth. Cool me off. It's a little bit warm. I don't know why I wore long sleeves. I guess I was cold when I got up this morning because all the windows were open overnight. But yeah, so what do cilia do? They have this back and forth movement where they create currents. They're creating currents outside the cells to move stuff along. Okay, is it moving the cell? Did I get out of this chair and move? No. The cilia are moving stuff along the outside of the cell. Another example of an organelle containing microtubules is the flagellum. The only cell in the human that contains a flagellum is the human sperm in the man, okay? And they are not creating currents. What they are doing, they're creating this whip-like movement so that the sperm is motile, sorry, kind of like a snake slithering along. By the way, that's the only cell in your body that can do that kind of movement because it's the only one that has a flagellum. Now, in contrast to that, we have another projection of the surface. So we have the cilia, which are plasma membrane covering, creating external currents. Then we have the flagellum, which move the sperm. Here, instead of moving your arms, it's like your fingers, okay? It's just plasma membrane and cytoplasm inside there, okay? There's no cytoskeleton, there's no microtubules, no microfilaments, no intermediate filaments. And what these do, each one of these would be called a microvillus. So altogether, more than one microvillus is known as a microvilli, okay? And what they do is they increase the surface area of the cell, okay? So they're only on the free surface of the cell. And so instead of the surface area being this much, now we have this much surface area. So anytime you hear about a cell having microvilli, you know that that cell is either going to be absorbing stuff or secreting stuff because it's increased its surface area. Just like anytime you hear about a cell having cilia, it's going to be creating those external currents. Okay. So a microvillus is not the same thing as a cilium. Structurally, they look nothing like each other. And the microvillus is only about 5% the length of a cilium. Sorry, so here is a cell. There's where the plasma membrane would be. Here are microvilli, and then here are cilia. And if you notice, you can see the cytoskeleton within the cilium itself. Now, the last organelle we're going to talk about is the mitochondrion. And notice I-O-N at the end for the singular form, not mitochondria with an A. That's the plural forms. And the mitochondria does have membranes, but it's not part of the endomembranous system. In other words, nothing comes to it and fuses, and nothing leaves and pinches off. It has an outer smooth membrane, kind of like a oval, like a jelly bean. Inside, the inner membrane has these foldings which have these shelves known as cristae, these infoldings. And the mitochondria is where 2 of the 3 parts of cellular respiration can occur to produce massive amounts of ATP. So places where we need a lot of ATP for that organ to work, like muscles, liver, you're going to have lots of mitochondria. And so on the right here is an example of a microscopic picture of a mitochondria. And so I want you to try to determine what type of microscope took that picture. And then also, can you figure out what organelle this is over here to the right? Here's a hint. It has all of these little dots on its surface, this other organelle here, over there. And so what organelle is this? What is this doing? And then what type of microscope for that? So if you have any questions about that, ask me. So I alluded to cellular respiration, so let me talk about cellular respiration for a second. If you're taking chemistry, you'd have to know the chemical equation. We do not have to know it. Basically, we do need to understand what's happening. Cellular respiration is where your body takes sugar and uses oxygen. And the whole point of it is to make energy. But in addition to energy, we are also producing water and carbon dioxide as waste products. So we get rid of the water, we get rid of the carbon dioxide. It goes out into the universe, and plants take the water and carbon dioxide, and they take energy from the sun, and they make sugar and oxygen, which then we consume. Okay. So in cellular respiration, there are 3 big steps to cellular respiration. The first step is called glycolysis. Glyco for glycogen, glucose, sugar. Lysis still means to cut something up. So glycolysis, sugar breaking. We are taking a 6-carbon glucose molecule and breaking it down into 2 3-carbon molecules, okay? That's going to require a little bit of energy to break it down, but we are going to produce a little bit of energy in this whole process. This process occurs in the cytoplasm, and we make 2 ATP. Well, we actually make 4, but it takes 2 to do the breaking, so we have a net of 2 ATP. Then, if there is oxygen, and only if there is oxygen, these 2 3-carbon molecules at the end of glycolysis will enter the mitochondrion. The first place they go is to the matrix, the fluid stuff inside the mitochondria, for the second step, which is known as the Krebs cycle or the citric acid cycle. In this cycle, they are making chemicals, which basically are going to have to do with electron carrying, so we're not going to learn about them for this class. But those structures then move to the cristae of the mitochondria. So it's showing a cristae underneath there, and that's where the third process, known as the electron transport chain, occurs. And in that electron transfer chain, this is where we are going to be making the vast majority of our energy. So if everything goes perfectly at the end of this, we can end up with 36 ATPs, but only if there was oxygen. If there was no oxygen, how much ATP did we end up with? Okay, so as you can see, there's a lot of moving parts in the cell. So we're done with our brief review of the cell. Read your textbook and ask me if you have any questions. We have one more video about the cell to talk about the cell junctions. Thank you for all your work.